This is our last section of chapter 5. It's going to be short, but very, very packed. Our previous portion of this chapter was about catabolism primarily. Now we're going to look at photosynthesis, what is photosynthesis, and how it is performed in certain types of microbes. Now, when we're looking at photosynthesis, we need to remember that this is going to be one of our phosphorylations. Remember, we talked about phosphorylation, the adding of a phosphate, in the previous portion of this chapter. And we've already covered substrate level phosphorylation and then oxidative level phosphorylation. And the last one of this phosphorylation in this chapter is photophosphorylation, which is going to be performed in photosynthesis, which is basically creating ATP, okay, by adding a phosphate to ADP, adenosine diphosphate, by the use of light energy. Now, let's first look at what is photosynthesis. Let me get my textbook opened up to chapter 5 so we can get to that section and what page we are actually on. That covers photosynthesis and the pathways. So photosynthesis starts on page 144, where it starts talking about it and the chemicals and structures and so forth. So let's first look at the term photosynthesis. Basically what it is capturing light energy from the sun and using it to drive the synthesis of carbohydrates from carbon dioxide and water. Let me say that definition again of photosynthesis. It's capturing light energy from the sun and using it to drive synthesis of carbohydrates from carbon dioxide and water. What types of organisms tend to do this when we're dealing with microbes? Cyanobacteria. See if I can spell that out here for you. Cyano, and that's an O, bacteria. I wish I had a regular whiteboard. Cyanobacteria, purple sulfur bacteria, green sulfur bacteria, green non-sulfur bacteria, purple non-sulfur bacteria, algae, green plants, and a few protozoa that are photosynthetic. Those are your organisms that are going to perform photosynthesis. What is going to be able to allow us to perform photosynthesis? There are certain molecules, pigment molecules, called chlorophylls. So kind of like the cytochromes we had in the electron transport chain, these are going to be specialized pigments that are going to be set up in a chain to be able to allow light energy to excite them to be able to create electron energy to run a gradient. There are different types of chlorophylls. Chlorophyll A, chlorophyll B, bacteria chlorophyll A, and these all are going to be slightly different and perform slightly different functions. Now, as far as what we need to know, there are chlorophylls. There's different types of chlorophylls, and they're all going to be designed to get excited, I should say, when light energy hits them. Now, where are these chlorophylls going to be? They're going to be in photosystems. And we're going to talk about photosystems a little bit more here in just a moment. Photosystems look very similar to the electron transport chain we looked at earlier. And they're going to be embedded, these chlorophylls, in these stacks, okay, called thylakoids. That's a D right there. Sorry, it's running out. Let me type, just say it again. Thylakoids. I don't know if it's going to write it, but let me erase that. So let me clarify what I'm trying to say. All right, this is thylakoid. And then they'll be in these stacks. All right. These stacks are called grana. I'll figure out how to get this to work eventually. So that is what our chlorophyll is basically going to be inside a thylakoid, and then the stack is known as a grana. We're going to talk about these just in a minute when we actually look at the photosystems. I'm trying to put this together for you, and we'll actually make some sense out of this. So this is all going to be our one little piece. Here it's going to go into our photosystems. But we need to understand those terms and understand what they are. All right, so now let's get to the actual main part of this. It's going to start a little bit of confusion. I hope it's not too bad, but we have two different things going on when we're performing photosynthesis. We're going to have one segment that we call light-dependent reactions, and these reactions, just like they sound, if I can spell things right, are dependent upon light. They must have light in order to perform. And then these are also known as energy fixing reactions. Same term, same thing, just a different term. Why are we calling these energy fixing reactions? Because this portion of photosynthesis is going to be responsible for creating the ATP that we need to further go into our next step, which is going to be to fix our sugars and create sugars to store. So we have to create energy in order to be able to harness and drive the work to put together the actual sugar molecules. So first things first, we must use light, get energy, and then go in to actually fix our sugars. So it's a little more complex than, hey, they get to fix their own sugar. Hey, that's a cool thing. So we have a little more complexity going on here. So we have what's called the light-dependent reactions that are going to be responsible for creating energy. Now, let's take a look at what we have going on here with the energy fixing reactions, are called photosystems. Now with photosystems, we have the first one, known as photosystem 2, and then we'll have our second set, which is known as photosystem 1. Let's take a look at what page this is on. It is on page 147, and you'll notice that these look very similar to the electron transport chain we may have seen earlier, especially the bottom one, which is non-cyclic photophosphorylation. So we're looking at the different forms of phosphorylation and also looking at the photosystems that are involved within them. If you'll look, you notice there's two green pigments at the bottom in non-cyclic photophosphorylation. In that one, you'll notice photosystem 2 is the first one and photosystem 1 is the second one. Now let's take a look at what's going on with cyclic versus non-cyclic photophosphorylation. And I've got some little videos that also go through this too, so we'll just kind of reiterate what's going on. With cyclic photophosphorylation, it's a mouthful, isn't it? What you have is basically photosystem 1 getting excited, and you'll notice how, so I put it photosystem 1, it gets excited and circles energy in a loop. Okay, I'm trying to make this as simple as I can. So light rays come down, hit it, excite it, and continue to cycle energy in a loop, continuously creating a gradient to create energy and harness it to funnel ATPase pump that you notice is going further down the line. That one's fairly simple. See if I can move this over a little bit. Photosystem 2, excuse me, not photosystem 2, my bad, non-cyclic sorry, I'm getting ahead of myself, phosphorylation is going to use photosystem 2 and photosystem 1, okay? So it uses both of them. This one to me is the most important one because it uses everything all at once and you can see the big picture. So let's look at photosystem 2, and this is on page 147, if I haven't already said so. I believe I just did, but let me go ahead and reiterate it. And you'll notice that at the very beginning, you start with a light ray coming down and hitting photosystem 2. All right, so the light ray hits photosystem 2. Now what happens? Well, up above, you notice that you have water, right? Okay, so we get light energy excites, let me try to word this in a way that makes sense. I keep getting ahead of myself. Light energy excites photosystem 2, all right? Then it breaks the bond in water. Isn't that so cool? I just think that's so cool. Guess where our oxygen comes from with plants? Right here, okay? It doesn't come in other ways that we may think. It doesn't modify carbon dioxide and get the oxygen. No, this is where it gets it, right here. It takes light energy, excites photosystem 2, and with that energy that it receives, it literally breaks the bonds in the water and creates that chemiosmosis hydrogen gradient we saw before in the electron transport chain. Now we're creating here to channel and control our ATP synthase pump here. Okay, so I just think this is so amazing. So I'm trying to keep myself from getting too excited. This is really amazing, really cool. Light energy literally excites photosystem 2, breaks the bonds in water, and creates the hydrogen gradient we need. See, I'm so excited I can't even type. All right, it creates the hydrogen gradient. All right, so there we are with creating that hydrogen gradient we need to build up chemiosmosis. All right, so it's going to continue, electrons continue to travel down. You'll notice they go to photosystem 1, get excited again. They're going to cycle back around too because we have cyclic photophosphorylation occurring at the same time. Okay, and it's going to continue. And you notice how we create NADPH because we're going to need that. And now, what is the ATP synthase pump going to create? ATP. Okay. Now, NADPH and ATP go on to the Calvin-Benson cycle. Now, this is going to be your light-dependent reactions right here. All your energy-fixing reactions, light-dependent reactions are this right here. What we're going to do now is I'm going to go ahead and show you a video because unfortunately I couldn't really draw some pictures. I'm using a different computer, so this is a little different. So I'm struggling a little bit with doing the pictures. So I'm sure that's not a disappointment to you anyway because I draw like a second grader. I will go ahead and show some videos for you on this, and then what we're going to do is go on into our light independent reactions. So we're actually fixing the sugars. So now we've created our energy. Now we're going to go and create our sugars here in just a moment. So at this point in time, we're going to go ahead and just go through a little overview of photosynthesis and make sure we understand what it is and what can perform photosynthesis.
Plants, algae, cyanobacteria, and other prokaryotes have the ability to use water and carbon dioxide to produce glucose. This process is called photosynthesis, from photo meaning light, as they obtain the energy to perform this feat directly from the light of the sun. Photosynthesis takes place in folds of the cell membrane in prokaryotes such as cyanobacteria. It's believed that such prokaryotes were ingested by eukaryotic cells, forming an organelle called a chloroplast. Inside the chloroplast are stacks of structures called thylakoids. The thylakoid membranes contain all of the components that participate in the light harvesting reactions of photosynthesis. The molecule that captures the energy of light is called chlorophyll. The light energy captured by chlorophyll is used to produce a proton gradient during the light-dependent reactions, and this proton gradient is used to produce ATP. In addition, NADP is reduced to NADPH during the light-dependent reactions. The ATP and NADPH produced during the light-dependent reactions are used to make glucose in the light-independent reactions, also called the dark reactions or the Calvin-Benson cycle. Plants, algae, cyanobacteria, and other prokaryotes have the ability to use water and carbon dioxide to produce glucose. This process is called photosynthesis, from photo meaning light, as they obtain the energy to perform this feat directly from the light of the sun. Photosynthesis takes place in folds of the cell membrane in prokaryotes such as cyanobacteria. It's believed that such prokaryotes were ingested by eukaryotic cells, forming an organelle called a chloroplast. Inside the chloroplast are stacks of structures called thylakoids. The thylakoid membranes contain all of the components that participate in the light harvesting reactions of photosynthesis. The molecule that captures the energy of light is called chlorophyll. The light energy captured by chlorophyll is used to produce a proton gradient during the light-dependent reactions, and this proton gradient is used to produce ATP. In addition, NADP is reduced to NADPH during the light-dependent reactions. The ATP and NADPH produced during the light-dependent reactions are used to make glucose in the light-independent reactions, also called the dark reactions or the Calvin-Benson cycle.
Now let's go ahead and go into our cyclic and non-cyclic photophosphorylation segments. This will be photophosphorylation with the cyclic pathway.
Photosystem 1 is composed of a large number of chlorophyll molecules, which harvest light and pass the excited electrons between the chlorophyll molecules until the excited electrons reach the reaction center. After the excited electrons are passed to the reaction center, these electrons are passed to the next protein in the electron transport chain. In the cyclic photophosphorylation pathway, the electrons that are released by the excitation of photosystem 1 pass through 3 sets of electron transport proteins, finally returning to photosystem 1. On their way, the electrons help establish a proton gradient as they pass through a group of proteins called cytochromes. The proton gradient in turn drives ATP synthase to produce ATP through chemiosmosis.
Now we're going to go ahead and take a look at the non-cyclic photophosphorylation, and we'll be done with that portion.
The non-cyclic photophosphorylation pathway utilizes both photosystem 1 and photosystem 2, and it produces the reducing molecule NADPH, as well as indirectly producing ATP. Unlike the cyclic photophosphorylation pathway, where the electron source and final destination are both photosystem 1, the electron source of non-cyclic photophosphorylation is water.
So you'll notice here, let's go ahead and take a look at this again as it's going through these stages. I'm going to kind of just fast forward through it. And we notice how water, okay, so sunlight comes down and excites photosystem 2, water comes up, and it splits the water molecule. When it splits the water molecule, we get oxygen released in the atmosphere, and hydrogens are now going to create a gradient for us to be able to funnel through the ATP synthase pump to create ATP. So now that we've talked about our light-dependent reactions, this portion is going to be our light-independent reactions, also known as carbon-fixing reactions. This cycle is the Calvin-Benson cycle. Now we created, I say now a lot, we have created energy from our energy-fixing reactions in the form of ATP and NADPH. These will now be used to form glucose. Let's take a look on page 148 at that cycle, and you'll notice that it looks a little bit like a backwards glycolysis, because that's kind of what we're doing, is a backwards glycolysis. So we're starting off with ATP, we're starting off with NADPH, that are our forms of high energy. We're going to burn them, we're going to expend them to be able to put together bonds, because when we put bonds together, this doesn't create energy. Instead, it uses energy when we put bonds together. So we have to have energy in order to create the glucose molecule from what we're going to be using. So you'll notice here, we start with carbon dioxide. That's what a plant will use. End product, it fixes the carbons to form glucose. So you'll notice here, I don't want you to understand every single thing that is going on here. However, RUBP is a fairly important term. That's ribulose biphosphate. We take that, add 3 carbon dioxide molecules, use energy, and as we go along, we're going to continue to build our molecule. You get toward the bottom of this structure, you notice there is what we call glyceraldehyde 3-phosphate. That's going to break off, you notice, essentially. We get 2 of those. We bind those together to form glucose. So it looks similar to what we were doing with glycolysis. Remember, we wound up with 2 3-carbon molecules. Well, there's 2 3-carbon molecules that get bound together to form glucose. So that's what we're basically looking at in the Calvin-Benson cycle. I'm going to show you a video that honestly is a little more complex than what I'm interested in you understanding from this. It is a light independent reaction. So this you might also hear the term of dark reactions. Why? Because here's a cool thing. This does not need light. So have you ever heard that plants need darkness sometimes? Yes, because they don't need sunlight in order to perform this. They've already created their energy during the light times and harvested their energy. Now what we do is what we call light independent reactions that can be performed in the dark, okay, that will fix carbon into glucose molecules. What do we need in order to do that? Well, we need ATP, we need NADPH, and we need carbon dioxide. All right, then we're going to go through and we're going to fix those carbons into glucose. We start off with ribulose biphosphate, and then we're going to have glyceraldehyde 3-phosphate at the end of this stage that gets bound together to form glucose. All right, so this is the independent reactions, also known as dark reactions, Calvin-Benson cycle, carbon-fixing reactions. They're all the same thing. I hope that you can hear the audio. If you can't hear the audio on any of these, let me know so I can go back and correct this and make sure you're able to hear these videos. I'm going to go ahead probably and go through the photosynthesis one just one more time again after this one is over. I shouldn't say photosynthesis one. This is part of photosynthesis. I'm going to go through the light dependent reactions and make sure you can hear the videos as well.
The Calvin-Benson cycle, sometimes called the light-independent reactions or dark reactions of photosynthesis, is the enzymatic pathway that uses the chemical energy of ATP and the reducing power of NADPH to generate glucose and other sugars from water and carbon dioxide. Through 2 rounds of the Calvin-Benson cycle, a single glucose molecule is produced from 6 carbon dioxide molecules and 6 water molecules, at the expense of 18 ATP molecules and 12 NADPH molecules. In the first step of the Calvin-Benson cycle, 3 carbon dioxide molecules from the air are covalently attached each to one 5-carbon ribulose bisphosphate molecule. The 3 resulting 6-carbon sugars react with water and break apart into 2 3-carbon sugars each, resulting in 6 3-phosphoglyceric acid molecules. This step is called the carbon fixation step. In the second step of the Calvin-Benson cycle, 6 molecules of ATP are used to phosphorylate each of the 3 phosphoglyceric acid molecules produced in the first step, resulting in 6 1,3-bisphosphoglyceric acid molecules. 6 NADPH molecules are then used to reduce the 1,3-bisphosphoglyceric acid molecules to 6 molecules of glyceraldehyde 3-phosphate, G3P. The ATP and NADPH required for these reactions are produced in the light-dependent reactions or through other catabolic pathways. After glyceraldehyde 3-phosphate is produced, 1 out of every 6 glyceraldehyde 3-phosphate molecules is diverted out of the cycle. When 2 glyceraldehyde 3-phosphate molecules have been diverted, one of them is converted into dihydroxyacetone phosphate. These 2 molecules react to produce fructose 1,6-bisphosphate, which can be used to produce glucose and other sugars. The reverse of these reactions occurs during glycolysis. 5 out of every 6 glyceraldehyde 3-phosphate molecules continue in the Calvin-Benson cycle to regenerate the ribulose bisphosphate required in step 1. 5 glyceraldehyde 3-phosphate molecules each contain 3 carbon atoms, for a total of 15 carbon atoms. Through a complex series of reactions, these 5 3-carbon molecules and 3 molecules of ATP are used to produce 3 5-carbon ribulose bisphosphate molecules, which can then proceed with another round of carbon fixation.
Plants, algae, cyanobacteria.
Plants, algae, cyanobacteria, and other prokaryotes have the ability to use water and carbon dioxide to produce glucose. This process is called photosynthesis, from photo meaning light, as they obtain the energy to perform this feat directly from the light of the sun. Photosynthesis takes place in folds of the cell membrane in prokaryotes such as cyanobacteria. It's believed that such prokaryotes were ingested by eukaryotic cells, forming an organelle called a chloroplast. Inside the chloroplast are stacks of structures called thylakoids. The thylakoid membranes contain all of the components that participate in the light-harvesting reactions of photosynthesis. The molecule that captures the energy of light is called chlorophyll. The light energy captured by chlorophyll is used to produce a proton gradient during the light-dependent reactions, and this proton gradient is used to produce ATP. In addition, NADP is reduced to NADPH during the light-dependent reactions. The ATP and NADPH produced during the light-dependent reactions are used to make glucose in the light-independent reactions, also called the dark reactions or the Calvin-Benson cycle.
Photosystem 1 is composed of a large number of chlorophyll molecules, which harvest light and pass
The excited Photosystem 1 is composed of a large number of chlorophyll molecules, which harvest light and pass the excited electrons between the chlorophyll molecules until the excited electrons reach the reaction center. After the excited electrons are passed to the reaction center, these electrons are passed to the next protein in the electron transport chain. In the cyclic photophosphorylation pathway, the electrons that are released by the excitation of photosystem 1 pass through 3 sets of electron transport proteins, finally returning to photosystem 1. On their way, the electrons help establish a proton gradient as they pass through a group of proteins called cytochromes. The proton gradient in turn drives ATP synthase to produce ATP through chemiosmosis.
So that was cyclic, I apologize, that was cyclic photophosphorylation. Now we're going to look at non-cyclic photophosphorylation.
The non-cyclic
The non-cyclic photophosphorylation pathway utilizes both photosystem 1 and photosystem 2, and it produces the reducing molecule NADPH, as well as indirectly producing ATP. Unlike the cyclic photophosphorylation pathway, where the electron source and final destination are both photosystem 1, the electron source of non-cyclic photophosphorylation is water.
Hopefully that explains photosynthesis to you well enough that it makes sense. If you have any questions, don't hesitate to let me know. And this is the end of chapter 5.